Can you guys hear me okay?
Yeah.
Okay, cool. Alright, hi everyone. I'm Charlene Chen from Smithtown High School East in New York, and I'm excited to tell about my project on the development of a urine RNA Aptasensor-based kit to detect oxidative DNA damage in outer space. As astronauts explore outer space, they are exposed to a harsh environment that puts the human body under extreme stress, resulting in many adverse side effects. For the purposes of my experiment, let's take a closer look at microgravity and radiation. These two major stressors can create DNA damage through many different molecular mechanisms. This is a huge concern, as it can lead to diseases such as cancer, conditions such as premature aging, and also concerns for reproductive health, which will be crucial as we seek to extend our duration in outer space. And thus, it is absolutely imperative for us to monitor the genomic health of astronauts in the most efficient way possible. Some of the current tools used to measure DNA damage are the high-performance and mass spec liquid chromatography, and also immunohistochemistry tools such as the ELISA. However, these tools are not the most ideal for measuring DNA damage in space. This is due to many other drawbacks, which are that they are tedious, time-consuming, costly, highly specialized, and bulky, all traits that can hinder the detection process. And thus, the goal of my project will be to develop a tool that will instead be simple, quick, cost-effective, easy, and compact. To do this, I'll be using aptasenses. Aptasenses are a combination of aptamers and ribozymes. Aptamers are oligonucleotides that can bind to target molecules with high specificity, and ribozymes are RNA molecules that have the ability to cleave themselves. Many research works have combined these two components together to create aptasenses, which initiate cleavage after binding to a target ligand. And thus, they are often used as regulators for gene expression and also biosensors. And thus, this is what I'll be using for my project. The target on my tool will be AOXO-DG. AOXO-DG is one of the most common products of damage by oxidative stress, and thus a major biomarker for oxidative DNA damage. A reminder that the conditions of microgravity and radiation, which exist on the ISS, are major sources of oxidative stress. They increase the production of ROS, or reactive oxygen species, which could inflict damage on our DNA. When ROS interacts with deoxyguanosine, it is changed into A-oxodeoxyguanosine, or AOXO-DG. And also, after ROS is scavenged, AOXO-DG is then excreted into urine, and this makes AOXO-DG an ideal candidate for our project, as urine samples are easy to isolate and are far less invasive as opposed to blood samples. For my aptasense design, it will be composed of a RNA aptamer discovered by Rink et al. With a high affinity for AOXO-DG. Additionally, there will be a ribozyme capable of self-cleavage, a ribosome binding site and anti-ribosome binding site, a SAN loop, and also a superfolded GFP, which will be our reporter gene. In this current state, the aptasense is not bound to its ligand AOXO-DG, and thus will not initiate cleavage, and the RBS and anti-RBS segments will stay together. This prevents ribosomes from binding to the RNA molecule, and thus no GFP will be produced. However, in the presence of AOXO-DG, the aptasense will bind to the molecule and initiate cleavage and a separation of RBS and anti-RBS. This allows ribosomes to bind to the RNA molecule and thus produce GFP. The aptasense will be produced from a DNA plasmid composed of the sequence for the aptasense, superfolded GFP, kanamycin resistance, and also an origin of replication. This is perfect for the BioBits tool, which is a portable and compact cell-free system containing all the necessary ingredients to make a protein from a DNA plasmid, and thus the aptasense will be created from the BioBits tool. Now let's move on to some of the experiments. For my freeze and fly, I will seek to determine the time it may take to produce a fluorescent signal, measure the level of detection of the aptasense, and also generate a standard curve for oxidative DNA damage. There will be 16 samples on Earth and also 16 samples on the ISS. 5 tubes will contain 1 microgram of the plasmid and also various concentrations of AOXO-DG. My negative controls will consist of a tube of water and a tube of AOXO-DG. I will also have a positive control containing a tube of just the plasmid for superfolded GFP. These 8 tubes will be duplicated for a total of 16. After, the results will be imaged using the P51 viewer for every hour for 10 hours, and also quantified using the Fiji imaging software. Now let's move on to what we may expect from this. So I expect that the ISS and Earth results will have a similar increasing trend of fluorescence as the concentration of AOXO-DG also increases. Additionally, a standard curve will be generated to compare the signal to the concentration of AOXO-DG. Ideally, the correlation will be clear, however, the fluorescence could also be at lower or higher levels. As a result of my Friesen fly, I will have determined the time it may take to produce the fluorescent signal, measured the level of detection of the aptasine, and also have determined the relationship between the fluorescence and the concentration. Now let's move on to the ambitious experiment. For my ambitious experiment, I will seek to test the functionality of the aptasine to accurately monitor oxidative DNA damage by comparing with known reliable methods such as HPLC. Additionally, it will also be important to test this in a mammalian model system such as a mouse. For the Earth portion of my ambitious experiment, I will have two groups of mice. The wild type will serve as our control, and the SOD1 double knockout, a mouse model engineered to accumulate ROS damage, will serve as the positive control. Urine samples will be collected from the mice for every 5 days for 35 days and split for two types of examination. The first will be using our proposed Aptasine, in which the urine samples will first be prepared in order to eliminate as much RNase and protease activity as possible. Then it will be added to a BioBits tube along with our plasmid. After, the results will be imaged and also quantified as previously mentioned. Secondly, the other urine samples will be prepared with solid phase extraction for the HPLC. The HPLC will allow us to confirm the level of AOXO-DG in the urine samples and thus determine the accuracy of our Aptasine. And all experimentation will be done in duplicate. For the ISS portion, again, we will have two groups of mice, with the wild type serving as our experimental, and the SOD1 double knockout, again, serving as our positive control. All the same procedures will be run with the Aptasine and the HPLC. The urine samples will be sent down to Earth for examination with the HPLC, and again, all experimentation will be done in duplicate. For each of the urine samples, we will have 5 tubes containing 1 microgram of our plasmid. And for my negative controls, I will have a tube of water and a plasmid, and also a tube of just urine. Additionally, we will have a positive control containing a tube of the plasmid of superfolded GFP, and again, these tubes will also be duplicated. Now let's take a look at what we may expect to see. For the wild type and the SOD1 double knockout on Earth, levels of fluorescence will not vary and instead be uniform with each subgroup, with the wild type having lower levels and the SOD1 double knockout higher. For the ISS mice, I expect to see an increasing trend of fluorescence due to the accumulation of genetic insults due to the space environment, with the wild type having lower levels and the SOD1 double knockout higher. As a result of my ambitious experiment, I will have determined the correlation between the Aptasine assay and also the HPLC results, and have evaluated the suitability of the Aptasine to monitor oxidative DNA damage in mouse urine samples. And the results of my ambitious experiment will showcase the successful implementations of the Aptasine-based technology. And the applications cannot be overstated. First, since our technology utilizes urine samples, a far less invasive process than a blood draw, it will be an easy method to detect DNA damage aboard the ISS. Additionally, it will also expand avenues for it will also lead to avenues for expansion, as numerous other kits could also be developed to monitor other biomarkers to monitor other human health concerns in space. And thirdly, this Aptasine-based technology also has applications right here on Earth. For instance, in low resource environments such as countries with few resources, labs with low funding, or even in classrooms where we can bring genetic studies to students who may one day themselves become future space pioneers. And lastly, I'd like to thank my mentor, Fran Navarro, my research teacher, Maria Zeitlin, as well as the GeneSpace team and the sponsors. These are my resources. Thank you.